bien de diffuser, c'est ça de diffuser. Très bon parfait. Et on fait subir une patient, il donne pourquoi je sais pas, c'est une patient, et là depuis 3 semaines, il dit ben, comment ça se fait que tu connais pas ce patient-là, que tu fais depuis 3 semaines? Le docteur, c'est une hiérarchie. Moi, je parle dans ma évaluation intellectuelle. On est jamais plus content dans quel territoire au Québec le territoire est correct.
Quelle université
Là, j'ai mis 53 pour c'est pas ce qu'il y a de non, t'as pas assez parce que
Good afternoon, everyone. So, before the questions from us to you, but what I'm going to do this year is I'm going to put a few questions that have been given in the past years, just that you have an idea of what kind of questions you should be preparing somehow. And so basically just remember that you will have 30 minutes per question. So I think, do you have any questions about the exam or about anything else? Or is it okay? Good. So today I'm very happy to welcome Martin Simard, who will be giving you the lecture on gene regulation mediated by microRNAs. So Martin actually is from Quebec, so he drove all the way to give you the lecture today, so you should be very happy, because also his research focuses on the understanding of how microRNA control gene expression, so he's really an expert in the field, so you should learn a lot. Just to mention briefly his CV, Martin is professor in the Department of Molecular Biology, Medical Biochemistry and Pathology at Laval University. And he's also the director of the Oncology Axis at the CHU de Québec, the research center of the CHU de Québec, So I think you're going to, this lecture is, I guess, a nice follow-up on the lecture we had last week on RNA biology, and I think you're going to learn a lot of exciting things. Thank you. Bye.
Good. Working? Yes. Okay, so I think this is maybe the 8th year that I'm giving this class here, so the class has changed quite a lot because it's one of the fields in biology that is moving quite fast. And today what I'm going to tell you about is pretty much, I want to give you a little bit of the background of where those microRNAs coming from. And this is quite interesting in the sense that you have to keep in mind that, in the 90s or something like that, it was not so obvious to think that small non-coding RNAs can do something in cells. People have been, focusing on messenger RNA or RNAs that are essential for protein production, or give proteins. And the discovery of small non-coding RNAs was quite shocking. And if you are interested in the history of science, you have to know that Monod back in the 50s, 40s has already thought that probably there is small molecules, nucleotides, that can regulate gene expression, which was quite impressive without having any experimental evidence. So the plan today, like I said, I will just briefly mention about the discovery of microRNAs. Also give you some example of function, and this is strongly biased through cancer because this is what I'm working on, but I'm pretty sure that in your field of interest in any fields of biology, you pretty much encounter some papers that mention about function of microRNAs in your favorite pathways or cellular pathways. I'll also tell you about I think one of the great assets of using microRNA, and especially as therapy, and there is different things that are currently in clinics that use microRNA at some point to either, detect some disease, human disease, including cancer, and also to treat some disease as well. Then after this, I'm going to switch to the more molecular biology aspect of my talk, which is where those microRNAs are coming from, how they work, and what we know about their function. And this is pretty much the main topic of my research group right now. So where those small RNAs are coming from, they're coming from a couple of studies that has been published in early 90s. So this is, for me, it's quite impressive, although we're coming to an end right now that I can say that most of you were born at this age, at that time, which, it's coming to be untrue now, but 1993 is it's very old. I mean, in a way that what we know about biology. So many aspects of biology was discovered by them, and, in the 90s, very few things. I mean, we fine-tuned many different pathways and cellular pathways and understand way better how the cells are working and how the biology in the cells works. But the fact of discovering an entire new regulatory pathway was quite impressive in this period of the year, so in the 90s. So how this comes from? It comes from this tiny animals that we call Cyanurhabditis elegans. So it's a tiny worm. It's 1 millimeter long, and this is what we're using in the lab, in fact, to do biology. So what is amazing about this tiny animals is, as you can see, it's transparent. So we can follow every single cell from the first division during embryogenesis till they reach adulthood. So it's a very good model to understand how development works and how development is controlled. And this is mainly what people are doing with this animal. Apoptosis has been discovered using this model system. So people are interested and still interested to understand how animals or cells can control very specifically developmental timing. And a way to use a worm or C. elegans as a tool is we can do mutagenesis, random mutagenesis. So just mutagenize the animal and looking at specific phenotypes. So one thing that you have to know to understand a little bit about microRNA discovery is you have to know the different developmental stage of the animal. So the animals lay eggs, and as you can see, this animal has egg inside the uterus that you're going to see right here. And what is also very exciting about this animal, in a way, that it's an hermaphrodite. So every single animal has the same genome. They have male gametes and as well as female gametes. So they can generate clones, and when you mutagenize them, you can after this look at your phenotype. And like I said, they lay eggs. Then after this, they have to go through very specific and detailed developmental stage till they reach fertile adults. So those two groups, so the Gary Ruvkun groups and Victor Ambrose's groups, have been working on this for many years. And one thing that they did is they mutagenized the animal and look what kind of gene or genetic lesion that can cause defects in the control of the development of C. elegans. And the genes that they found, they call it LIN4. So in worm, it's quite simple to understand what the genes means. It's the three-letter means for the phenotype. So LIN means for lineage defects. And what they observe is, in fact, LIN4 genetically suppress two genes, LIN14 and LIN28. And when they started to find out what LIN4 was coding for, And this is where they make, a significant discovery. So one thing that they discovered is the gene does not code for any protein. So the product of that genetic region in the genome was not giving any significant large RNA molecules that can, produce a protein or sufficient amount of amino acid to generate a protein. So this is one thing that they observed. They also observed by Northern blotting that in fact the product of the gene was a 21 nucleotide long RNA molecule. So this looks quite, from now looks quite normal, but it was something very intriguing because at the time we were thinking that all small RNA molecules were basically caused by degradation of long messenger or long RNA molecules. So it was quite clever to focus on this and think that maybe those tiny RNAs can do very something. And the other thing that they observed is when the gene LIN4 was expressed, so when they saw the 21 nucleotide long RNA, what they were observing is LIN14 level protein as well as LIN28 was going down. So when the 21 nucleotide RNA was going up in expression, the protein of LIN14 and LIN28 that were genetically shown to be silenced by a LIN4 was going down. And the other thing that they observed, so they look at the sequence of this 21 nucleotide long RNA, and they saw that indeed this small RNA molecules was complementary in part with the 3' non-coding region of LIN14. So just by looking at the sequence by hand, at that time they didn't have any computer analysis to do such a thing. So they saw that you have a quite good level of complementarity. So those two papers has been published in Cells in 1993, and in fact it didn't bring that much wow in the RNA field because people were thinking that maybe it's just something that those worms are doing in fact. So it was not really clear that it was a clear or an important regulatory pathway. And so I think what shapes the field in a sense, or started the field like that became a very major important field in gene regulation was the second paper from the Gary-Rufton group, where they discovered that those small RNA molecules were also found in other species, and especially including humans. So they cloned and they identified another microRNA or another small RNA called LET7. And what they saw is LET7 was within an RNA molecule, which was a little bit longer than the small one, so 21, and this is 70-ish nucleotide long. And what they saw is it formed a secondary structure with the other part of the RNA that has been transcribed by the polymerase II. And this structure, the stem loop structure, was conserved also in evolution. So it was also found in fly genome and as well as in human. And more interestingly, they also found that the LET7 microRNA, which was complementary to LIN41 3' UTR, this complementary with the 3' UTR region of LIN41 was also conserved in evolution. So all this was somehow suggesting that those small, at that time we were calling them small temporal RNA because they were expressed during development, was somehow a very important way to regulate gene expression, basically based on the fact, strictly based on the fact that they are conserved. So at that time, people started to try to clone those or to find more examples of small RNA molecules. And in fact, in 2001, so it's 14 years ago now, they have found or they have been, characterized about 100 of them, and this in different species. So in worm, we also found them in human cells and as well as in fly and plant, etc. So from now on, from 2001, it's basically where the microRNA field started. And this, in fact, has been named the breakthrough of the year at that time. So people were very excited about the discovery of those small RNA molecules, even though at that time we knew only 2 microRNA that has a real function, a biological function, which is LET7 and LIN4. So far, as far as I know now, we are over 10,000 microRNA that has been discovered, and this is, from different animals and plants. Basically, what microRNAs are doing is it's pretty limited what we know so far about them. But there is a trend there. So what we see is mainly associated to development, which is proliferation and differentiation of cells. So many function that has been attributed to microRNA so far has been associated to somehow an aspect of animals or plant development. So it goes to differentiation of neurons, proliferation, like I said, conferring apoptosis, morphogenesis in leaves and flowers in plants, and also in different aspects in vertebrates, so brain morphogenesis, hematopoiesis, and different tissue development. So to do this, the expression of microRNA has to be extremely precise. This is a good example of how the expression of microRNA can be very specific and tight. This is a mouse brain where people have done staining of different microRNAs, and as you can see here, they are expressed in very specific cells and tissue in the brain. Another landmark work that we know microRNAs are playing is during hematopoiesis, so during the production of those cells and differentiation of those cells. You can see that at many stages you will have subsets of microRNA that will be important for controlling the differentiation, the proliferation of those cells. So when we discovered all those microRNA, one obvious question that we have to address is what is the functions of microRNAs? So people have tried to make knockouts or mouse knockouts or any other types of animals, including worm knockouts of microRNA, and see what they do. And you can find out many things, but as far as we know so far, there is not many microRNA that give a clear lethality in the animal. So there is very few examples of this so far. Why we think it's happening is we think there is redundancy in the function of microRNA. So most of microRNA work as a group. I'm going to talk about it later on during my lecture, but this is, I think, what's going on right now. So we need to combine many of them to see a phenotype. But there is still some that give a very striking phenotype, like this microRNA miR1.2 that leads to overproduction of muscle cells, and in mice this is lethal, so the embryos cannot go through. There is also this is one of the famous clusters, so we call it the miR1792. Within this region, you have 5 different microRNAs, and when you knock out or you remove those microRNAs, you have a clear embryonic lethality in mice, suggesting again, that microRNA has very important function in biology. With regards to cancer, right after the discovery of a couple of hundreds of microRNAs, so back in 2001, people already started to look if there is any disease that can be related to microRNA. And this has been pivotal for people working in leukemia, because at that time we knew some chromosome rearrangement or chromosome aberration that has been found in different patients that were, the clinicians were unable to explain what's going on, because in the region that was affected, they didn't find any coding genes. So no proteins was produced from that chromosome spot. So they started to map the microRNA that we knew at that time. So this is back in 2004, and they basically discovered that the rearrangement that we were seeing in some patients suffering of leukemia, it was in fact enriched with microRNA molecules, so encoding for microRNA. So this was not a clear indication that microRNA were associated to cancer, but was a good correlation, or a very interesting correlation based on a leukemia patient. So this has been spearheaded by the Crochy group back in 2004, and he has been working on this for many years. So now we know many function of microRNA associated to cancer. So they can regulate their expression. So different oncogenes and super suppressor, like P53, P21, so the classical one that we know associated to tumorigenesis. MicroRNA can also be regulated by tumor suppressor and oncogene. MYC, for example, regulate the expression of many microRNAs. So it seems that we have an interplay between microRNA and classical gene associated to cancer. So microRNAs are a part of this chain of reaction that can lead to tumorigenesis. MYC is also a good example of it because, in fact, MYC is associated to the control of many different sets of microRNAs. And people now think that, besides affecting other protein coding genes, the fact that MYC can also affect specific expression of microRNA can be one of the key leads for leading to a key function to lead to the tumorigenesis. So there is also very intriguing things that occurs during the past years. So one thing that we know about regulate microRNA expression, and this is an example of this, miR34. So here, P53 regulates the expression, the transcription of miR34, and miR34 regulates, very key component in cells proliferation and apoptosis. So BCL2 and CDKs. But what was extremely surprising is the fact that also P53 can regulate microRNA, but post-transcriptionally, which means that P53 binds to a complex that we will then talk later on, and this, the association of P53 with this complex stimulates the production of subsets of microRNA. So now it's a function for P53 which is not transcription associated to transcription, but it's rather associated to the production of okay? And this was quite exciting for the field. And now we know that P53 and P63 can act at different stages during the production and the maturation of microRNAs that we're going to talk later on. But just to think about, you can quickly think that you can affect transcription of microRNA, you can affect also the production of microRNA, like I just briefly said, and they can also affect the expression of other genes which are associated to microRNA production and function. So there is many ways P53 interacts with the microRNA pathway. We were talking, like I said, about P53, which was known to be a bona fide transcriptional factor. Another gene that was quite also exciting to discover, so this is quite recent, 2012, where we show that, people have shown that BRCA1, so BRCA1 is another key player associated to DNA repair, but in this case, BRCA1 played pretty much the same role as has been discovered for P53, so it regulates the biogenesis of microRNA, of some specific microRNA. See, so we have now at least two DNA binding proteins that are associated to the production and maturation of microRNAs. And you will see later on why it makes sense. Just want to throw it there. So, if microRNA are really associated to cancer, you might think that the overexpression of some of them can lead to tumorigenesis. So, in a way, microRNA can act as an oncogene. So that was, and still for me, the best example of this, and this is miR10B, and its association with breast cancer. So what this group has shown, so in fact, the overexpression of this single microRNA, so miR10B, is sufficient to increase angiogenesis in tumors, okay? And it induces as well the metastasis formation. So overexpressing a single microRNA will lead to those key features in production of metastasis. And the other thing that they also observe is women that were suffering from breast cancer. So the ones that were metastasis free, so that they were not having any metastasis produced from their cancer, they were very low. The cancer cells were really low in Mir10B levels, and the ones that their cancer has been able to form metastasis has very high level of Mir10B. So Mir10B is linked somehow to the production of metastasis from breast cancer. And there is now more and more example of this associated to Mir10B. So Mir10B became a very good, interesting target to block or to suppress metastasis in breast cancer. One thing also that has been described a few years ago is that some tumor can become addict to the expression of some microRNA. What I mean about this is some tumor, like this is a very striking example. When you overexpress one single microRNA against Mir21 in the hematopoietic system, it leads to lymphoma formation. And if you shut down the expression of microRNA, what you see is the tumor shrunk very rapidly. Okay? And this somehow is an indication, again, that a single microRNA can act as an oncogene. So the overexpression of a single microRNA is sufficient to lead to a tumorigenesis. There is also an example where microRNA can act as a tumor suppressor, so which means that their natural target can be oncogene, and in absence of microRNA, what will happen is they will generate cancer cells. The first example of this was this microRNA that I told you about briefly, the LET7 microRNA, so the second microRNA that has been discovered, and the first one that has been discovered in humans. What the Frank Slack group have shown back in 2005 is, in fact, the level of microRNA was correlating with lung cancer. So if you take tissue coming from lung cancer, what you see is the expression of LET7 is very low. And one of the targets of LET7 is RAS, so the oncogene RAS. And when you have a very low level of LET7 microRNA, what you see is a significant level or increase of the RAS proteins compared to the non-tumorigenic tissue next to it. So it's a correlation that LET7, and they have also shown in this paper molecularly that LET7 regulates the expression of RAS protein, and this correlation causes cancer cells production. Another example of this is again in breast cancer. So I told you about Mir10B that act as an oncogene, and there is also microRNA in breast cancers that act as suppressor, work as tumor suppressor. So what they do is, if they re-establish the expression of two microRNA, Mir335 and 126, what they see is a significant decrease of metastatic area in the lung. It's a mouse model that generates metastasis in the lung. I found a xenograph of breast cancer. And what they have shown is, in fact, Mir335 control cell migration and Mir126 control cell proliferation. So again, we're coming back to the same type of function, which is tightly regulation of cell homeostasis by microRNAs. And now there is more and more example of this. So and it's pretty much the same way it's working. So you can, like I said, delete the microRNA, the region, the locus that codes for microRNA. So this is something that you see also in other genes. You can have also epigenetic silencing of microRNA locus. This is something that you can see, which is quite specific to microRNA. So you can have point mutation within the microRNA sequences, or the stem loop structure that I briefly showed you with LET7. And this, what will cause is a defect in the production or in the processing of microRNA. You can have genomic amplification, translocation, as you see with other genes, loss of epigenetic silencing. Point mutation in oncogenes. So microRNA binds by complementary sequence to their target. So if you have mutation in this binding site, it will affect the efficiency or the efficacy of microRNA to regulate gene expression. And this is quite important because, as you know, with even now with the high throughput sequencing, we're still focusing on the exome, so the region that codes for the protein. So after the stop codon, we don't really pay much attention of mutation that can be found there. And there is more and more examples that in fact, if you affect in the 3' non-coding region of a messenger RNA the sequence and you affect somehow the regulation by microRNA, that could lead to very significant aberration in the gene expression. And another thing which is also remnant to the discovery of microRNA is we also have to be careful about rearrangement, not only of the coding region or the promoter region that can affect the expression of a gene, but also the 3' coding region. Because again, if you swap or change this region, which is a place where microRNA mainly act in animals, so in the 3' non-coding region, but you will cause somehow misregulation of the gene. So now in the field we call those microRNA associated to cancer oncomeres. So this is either they are acting either as oncogenes or tumor suppressor that can somehow affect or lead to cancer cells. And there is a more and more way that has been discovered in the last couple of months and just last year, a way that microRNA can lead to cancer. Again, I briefly mentioned some of them, but you get when you change the balance between the targets of microRNA and the microRNA level, so by overexpressing just a little bit of the gene, you will just affect the balance of regulation that can be caused by microRNA, because as you can think, it's one microRNA that binds one messenger RNA. So if you can affect this function just by slightly increasing the level of messenger RNAs that are targeted by microRNAs, you can also change somehow the combination of genes that are expressed in the specific cells. MicroRNA binds several targets because it's complementary sequence to their target. It's not fully complementary, so they can bind different RNA molecules and affect its gene expression. So if you change this balance by starting to express a new genes or a new messenger RNA in the cell that can now be a target of microRNA, it can somehow change the balance of regulation that microRNA normally do. And a field that is currently actively studied is the role of microRNA in metastasis formation. So this review just briefly mentioned where at different stages you have microRNA that can affect different steps of metastasis formation. Again, just showing or just to give you a brief overview that microRNA can act at different places. So this is just a few examples of microRNA in cancer. As you may think, one of the key problems that we have in cancer is to detect the cancer first as soon as possible, and as well as to use the appropriate therapies for specific cancer. And quite rapidly, microRNA has been subject to those potential use as a biomarker, because it's a small RNA molecule that it's very easy to detect by standard system like RT-PCR, Norton blotting, etc. Our FISH analysis. So people have rapidly looked at microRNA level in different diseases, including cancer. That's the example that I'm giving you here. And just by looking at the expression of few hundreds of microRNA, even less than 100 microRNA, you can somehow classify different types of cancers just by looking at the expression of less than 100 microRNA, different microRNA. So this is very powerful because if you try to look within a huge group of messenger RNA or protein that is expressed in a single cell, so it's thousands of targets or thousands of messenger RNA or proteins, you have to find out within all this, big expression of a single cell, which combination can lead you or can help you to detect the cancer cells or early cancer cells and so on. So the fact that we can use a small subset of biological molecules like microRNA is very promising because in fact it's very contained and it's way easier to find out if it's really relevant or not. So many groups now and companies are working actively on this. And this is, I think, one of the best indications because usually in this case people are just taking two different type of cancer cells or tumors and different type of controls, and they knew already right up front that they have tumors in healthy cells, so it's kind of a bias the way you can look at it. But in this study, what they did is they looked, in fact, on only 48 different microRNAs expression level, and blindly they were able to detect for more than 90% of the tissue that they were observing the level of those 50-ish microRNAs. They were able to classify where the cancer cells were coming from. So they were looking at metastasis. So they have different metastasis the tissue, and they were just looking at the expression of 50 microRNA, and they were able to say this is coming from ovaries, this one is coming from breast, colon, and so on. So this was a very powerful study that really, I think, proved the potential of microRNA as biomarker. And another striking feature of microRNA is they are found in different biological fluids. And this is for me, it's quite amazing. So you can find some microRNAs in tears, and there is a very specific, for example, signature of microRNA that you can detect in tears. You have different biological fluid, breast milk, plasma, saliva, and so on. So people have been looking at this, and what we're thinking right now is, it could be very easy just by taking those fluids which are quite accessible, and anywhere in the world you can just look at microRNA profiling and find out if there is something going on in the body of this person, because if you see that a microRNA suddenly disappear, the level of expression is sky to the roof, I think it's a very promising approach that we're going to use in medicine very soon. So another thing with regards to microRNA is, for some reason which is not really understood, is they are extremely stable. So you can even use tissue that has been embedded in paraffin for 10, 20, 30 years, and you can still recover the RNA, small RNA from it, and detect microRNA molecules. And this is something that you can clearly not do with long messenger RNA because they become degraded quite fast. So they are extremely stable. We don't know much why, but I mean, I think it's again another aspect or feature of microRNA that makes this molecules a lead signature for different type of disease, including cancers. This is just a survey of different microRNA, specific microRNA that has been associated to different diseases, including cancers, and where you can detect them. So as you can see, many of them are in serum and plasma. This is usually what people are doing to look at biomarkers. But now that we know that we can find them as different fluids, that people are now looking at this carefully and see if we can use this even more efficiently. And this is also a field with regards to how you can find microRNAs in fluids, for example, in plasma and in blood. This is something that we don't know much why they are stable, why they are there. You can find naively that maybe it's just cells that breaks and provide microRNAs, but there is more than this, and this we know that. So And what has been found in the last few years, and they can be associated to different things. So the exosomes, if you know a little bit about that field, it's a very emerging field right now. So we have a lot of hopes about exosomes. So this is those I don't know how you call it, but not granules, but those small complexes that are expelled by cells, different types of cells, and they can go in the fluid, like in the blood. And we know that microRNA are really rich in this. We know also that they can be associated to HDL, so this lipid droplets that you find in the blood. You can also detect microRNA in there. We don't know really much how they can go into that kind of molecule or complex. They can also be associated to different type of proteins. And with regards to this and the fact that the host cells can just expel microRNAs, and those microRNA can somehow go into the blood and affect a recipient cells. That has been there for some years now, and people think that it can be a very exciting field to look at. And just this week, I just popped up this paper that has been recently published in Nature. And what they have shown is it's very striking. So we have seen that some type of cancer can generate metastasis in the brain, and they didn't know much quite how it works and things like that. But what they have seen is in fact is glial cells in the brain can generate a specific microRNA, that is expressed in the brain. And when the cancer cells arrive to this region in the brain, PTEN is suppressed in the cancer cells. And the fact that you block PTEN, you start, the cancer cells can proliferate and form a metastasis. And they very beautifully shown in this paper that this is specifically associated to the expression of one microRNA that is found in the exosome. So they are produced by glial cells. They go into the cancer cells, shut down PTEN, and now the cancer cells can start to be proliferate and form a metastasis. And this is quite striking to me. So now, for me, it's a clear proof that circulating microRNA in the microenvironment itself, this is a place where microRNA can act and relate to. So why microRNAs can become very powerful into a small subset of microRNAs? And like I said, it's simpler to identify good biomarkers from a set of 200 of microRNA, even less than 100 microRNAs. And if you compare it to maybe 40,000 messenger RNA that can be expressed in the same cells. They are extremely stable. Again, it's not clear why, but it's small RNA molecules that they are extremely stable. And what is really nice also about microRNAs is they can allow retrospective analysis of cancer tissue, so we can go back in our tissue bank and different type of banks that biobanks that you have in clinics and look very easily at microRNA levels and find out if you can find a trend or any information by looking only at microRNA expression. And I think this is one of the most important thing about microRNA, because as you may know, if you know a little bit about clinics, you have to have a way to look at your biological molecule quite efficiently and easily in the clinics. And this is the major problem with proteins, because you have to generate specific antibodies for each of the markers that you want. Sometimes it's like cumbersome to use those antibodies and be able to have a clean data set. And microRNA, for example, like I said, you can use conventional techniques that are already in clinics, like quantitative RT-PCR. It's very easy to do. You have probes now for every single microRNA that are expressed in human cells that you can easily do. You can, like I said, do staining just by fish, again, by using tiny probes that are available for all my human microRNA, very standardized and standard way to look at an expression in a tissue. So it's very easy to work and study and look at the levels of microRNA. And the tools and reagents are very easy to generate. So this is, I think, why microRNA have been pushed very quickly in clinics and trying to find out, in diagnostic kits and things like that. It's quite amazing. You have to think about this, that, all this has been discovered in early 2000, and we're already there. So there is kits in clinics that people are using and so on. So it's very, very fast. And it's not only associated to cancer. So this is, again, just to briefly mention, different types of disease, human disease, viral infection, mini viral infection, encounter and use microRNA in different ways. So viruses can encode microRNA that target viral microRNAs and regulate, for example, the viral life cycle. Viral microRNA can suppress expression of host genes, again, to favor his life cycle. Could be the reverse too. So viruses can produce proteins that block the expression of specific microRNAs and favor again its propagation and so on. So different ways you can think that viruses can use this microRNA pathway and affect gene expression. One of the first function of microRNA and small RNAs has been discovered in plants, where they show that suppressors of different microRNA molecules is very important to allow the viruses to infect plants. They may be important for gene imprinting. So this I wrote maybe because there is some evidence in the literature that you can find where microRNA can regulate chromatin structure and things, but I have to say it's not really clear yet, but I think there is something going on right there. And there is also different type of disorder, disease that you can look in the literature and you can easily find a microRNA somehow that has been related to that. The direct proof of them is not always clear, but I think it's going there as well. And if it's a very important regulatory system in cells, and this is a way biologists are thinking, is if it's really essential, most of the time viruses somehow evolve to use and to block and to affect this pathway, regulatory pathway. And there is more and more example of this. I just briefly mentioned you about plant viruses, and this is a human viruses, and in this case what this virus does is just express the long RNA molecules, and this long RNA molecules will interact with host microRNAs, so somehow it sequesters microRNAs from the host cells, and the sequestering of microRNA will lead to a misregulation of specific genes that are under control of microRNA. This again can favor the expression of the virus. So I told you about microRNA biomarkers. There is also the use of microRNA as therapies. I show you the beginning experience, some papers that show the expression of MIR10B, MIR21, that if you can regulate specific microRNA levels, you can somehow affect cancer progression. This is the major companies in US that works on this, trying to make tools that use microRNA or regulate microRNA expression and push it into clinics. And from different disease. There is different ways you can regulate gene microRNA expression. So one of the most obvious one, it's microRNA mimics. So what it means is you just generate a small RNA molecule that contains the exact same sequence of your specific microRNAs, and this can be coupled to different type of chemistry, lipids, and so on. And this way you can increase very rapidly the level of a specific microRNA in the cell. You can also obviously express microRNA from different type of vector. This is a viral vector. You can also, like the system that I show you about these viruses that hijack or sequester microRNA, you can do the same thing in cells as well. So if you overexpress RNA molecules that is fully complementary to microRNAs, to a specific microRNA, you can sequester microRNA from its targets. You can also use antisense oligonucleotides, so oligo sequences that are fully complementary to a microRNA, sequestered this way the microRNA molecule. So it's a way to block microRNA. We call commercially people are calling them antagometer. So this is kind of tools reagents that you can buy. There is also small molecules inhibitors that you can use that can affect different stage of microRNA production. This I have to say that it's not very common. Most of the times it's mimic that you see in antisense, and there is also this, which also not really common, where you use a small RNA molecule that will basically mask the binding site of a specific microRNA on a target. And again, this is not really used right now. It's more the mimics and the antisense that you probably have seen as well. So is it really working? So there is proof of principle to be done. Obviously, the first one you have to do is an animal model. So this work has been done in mouse. And what they shown, so they focus on this one on miR26A. So we know that miR26A is decreased in liver tumor, as you can see here. So this is coming from patient tissue. And what miR26 is doing is, in fact, is controlling cell cycle. This is what miR26 is doing. So if you remove miR26, there is no cell cycle control, and cells can start to proliferate, and it will generate the cancer cells. And so what this group did is they express miR26A in the liver cells, in the liver of mouse liver. So they use this retroviral system, vector system, where you can have the miR26A here, and you just started to express massively miR26A in liver cells. And this is coupled to GFP to show that the infection works pretty good. So this is just inject in the vein tail, just in the bloodstream, and go directly to the liver. Obviously, liver is an easy target because everything's go there when you inject in the blood. But regardless, I mean, you can do it very efficiently. You don't affect other microRNA levels, and this is something which is very important. And this is the effect that they see. So this is injected with a control vector, so you can see a lot of tumors going into the liver. And when you inject the viruses that express miR26A, what you see is a complete regression of the tumors. So suggesting again that overexpressing a microRNA or reestablishing the level of a microRNA in a tissue can suppress the formation of cancers and tumor cells. So this was quite striking. And they showed basically that by doing this you somehow recovered the control of cell cycle. So that was the reason, the mechanism behind the effect of miR26A. So there is more and more examples that you can find. Obviously, most of them are in the mouse model. There is some clinical trials right now that are going in human. I know that there is one drug based on microRNA that is associated, right now in phase 2. So it seems to work. They're well tolerated by humans, and there is obviously has to be. Right now, people are treating very specific disease, viruses, infection. This is some of them are pushing clinical trials right now. But there is also hope because, as you know, when you want to generate a drug that specifically target cancer cells, for example, it's very tricky. I mean, liver, like I said, it's an obvious target, everything going there. So, I mean, you can easily target liver, but if you think about other places or other tissues where cancer grow, you have to be able to specifically target them. And last year, in fact, the Frank groups used to be at Yale, now is at Harvard, came out with a very clever strategy. So the nice thing with microRNAs and antisense, and it's very tiny molecules that you have to generate. And this is something that it's very interesting because the major companies in U.S. that works on this, trying to to make tools that use microRNA or regulate microRNA expression and push it into clinics. And from different disease. There is different ways you can regulate microRNA expression. So one of the most obvious one is it's microRNA mimics. So what it means is you just generate a small RNA molecule that contains the exact same sequence of your specific microRNAs, and this can be coupled to different type of chemistry, lipids, and so on. And this way you can increase very rapidly the level of a specific microRNA in the cell. You can also obviously express microRNA from different type of vectors. This is a viral vector. You can also, like the system that I show you about these viruses that hijack or sequester microRNA, you can do the same thing in cells as well. So if you overexpress RNA molecules that is fully complementary to microRNA, with specific microRNA, you can sequester microRNA from its target. You can also use antisense oligonucleotides, so oligo sequences that are fully complementary to a microRNA, sequester this way the microRNA molecule. So it's a way to block microRNA. We call, commercially, people are calling them antagomeres. So this is kind of tools and reagents that you can buy. There is also small molecules, inhibitors that you can use that can affect different stage of microRNA production. This I have to say that it's not very common. Most of the time it's mimic that you see and antisense. And there is also this, which also not really common, where you use a small RNA molecule that will basically mask the binding site of a specific microRNA on a target. And again, this is not really used right now. It's more the mimics and the antisense, or the FC as well. So is it really working? So there is proof of principle to be done. Obviously, the first one you have to do is in animal model. So this work has been done in mouse. And what they shown, so they focus on this one on miR26A. So we know that miR26A is decreased in liver tumor, as you can see here. So this is coming from patient tissue. And what miR26 is doing is, in fact, it's controlling cell cycles. This is what miR26 is doing. So if you remove miR26, there is no cell cycle control, and cells start to proliferate and it will generate cancer cells. And so what this group did is they expressed miR26A in the liver cells, in the liver of mouse liver. So they use this retroviral system, vector system, where you can have the miR26A here, and you just started to express massively miR26A in liver cells. And this is
Again, another transcription factor, SMAD, as you may know. It's also important for maturation of specific microRNA. And this is why I was telling you about that, makes sense that transcription factors is as well associated to maturation of microRNA. Because microRNAs within their biogenesis, they have a stem loop structure molecules that is associated. So a stem loop RNA or DNA molecule, it looks pretty much the same thing. So most of the DNA binding proteins recognize the major and the minor groove of the double helix. So if you produce a double helix with RNA instead of DNA, the same type of protein can recognize major and minor groups. And this is why I think we'll have more and more examples of this where classical transcription factors can also act on microRNA processing because of the specific molecules that is generated during their biogenesis, which is the stem. There is also HNRNPA1, which is a classical RNA binding protein associated to splicing and so on. Again, that can be very specific for biogenesis of microRNA. From all the examples that we know so far, is the biogenesis of microRNA is controlled by specific proteins and only a subset of microRNA are sensitive to this. So it's also explaining or promoting somehow that the stem loop structure, the sequence itself, is very unique from one microRNA to another, and this will drive somehow their biogenesis. So I think we're just starting to see how it works, and for doing those kinds of studies, we have to look at very specific microRNA. And this is just to briefly show you, a different stage where different proteins can act. Most of them are affecting the processing of primary and precursor microRNA. And this is the one that we see. Obviously, because it's expressed by RNA pol 2, it's also subject to specific regulation of transcription of microRNA. This is a field that that people are naturally studying right now, not as much as we should. And so again, there is many ways during the regulation, different step of maturation of biogenesis of microRNA that you can somehow regulate. Another thing that we started to look at, and this is one of the work that we have done in the lab, is good, you can generate microRNA. But if microRNAs are stuck there and be able to regulate genes somehow forever, it can be deleterious for the cell because you need someone, because it's so tightly regulated during biogenesis, we can expect that once they have been used, they have to be somehow turnover, degraded, and be able to stimulate their degradation. And in fact, a few years ago, we discovered a complex that is important for degradation of microRNA. It's called the DCS1 hexavalent complex. And so what's happening is you have this microRNA associated to the argonauts, blocks the expression of a target, and once it's done, you need to recycle argonauts, and you need to degrade microRNA. Because like I said, if you want to make sure that you control the biogenesis or the production of microRNA, you have to get rid of the microRNA that has been used. So this is what this complex is doing. So it's just degrading microRNA and favors the recycling of argonauts, which we know is a very limiting step. And this, we have originally shown it in worm, and now we know we just published a paper a couple of days ago where we showed it's also 3 unit cells. So this complex itself is also important for microRNA turnover itself. Another way to regulate microRNA. So an example that I show you is affecting biogenesis, their production somehow. It can also affect or change their sequences. And the cells has a way to do it. To change RNA molecule sequences, it's called editing. So there is RNA editing in the cell. That's been known for many years for ribosomal RNA, SNO RNA, but now we know it's also true for microRNA. So this is a type of enzyme called ADARs, and what they do is they change the A for an I, and when you change an A for an I nucleotide, it changes completely their base pairing with another nucleotide. So A paired with U, I will pair with the G. So change swap their binding. So it's alter the base pairing, like I said, and also can alter the stability. So this is the enzyme responsible for this, ADAR1 and ADAR2. And we have seen that ADARs can affect many things. So if the editing occurs at the primary microRNA, so the structure or the molecules produced after transcription, this can inhibit the processing of microRNA molecules. So either the Drushel microprocessor complex or even the dicer the cleavage, and this can favor the degradation. In some cases that has been reported, but the addition is essential to promote the maturation of the biogenesis of microRNA. This way, the changing in the nucleotide change the structure, the way the precursor, the primary or the precursor looks like, the stem loop, and this will favor the production of microRNA. And because you act on the nucleotide sequences, you can also affect their specificity, again, like mutation that I told you about. Another thing that's starting to emerge as well in the field, and this is again a very recent paper, I told you about this Lin28 that recruits a SATase enzyme that adds use to precursor microRNA and stimulate degradation. So this is a way to do it, like you see here. But now we know also that this activity can be also important to make a proper maturation of microRNA. In this case, there is subsets of those types of enzymes, okay, that will add only one use at the end of the precursor. And this addition of one use can stimulate the degradation, or not the degradation, but the biogenesis instead of two. But we know that the cells will have, through this SATase enzyme, will have a single nucleotide, a U to it, and the addition of a single U will make it two nucleotides overhang, and now it can be, processed by dicer and so on. So this is also, again, something which is really new that we're starting to explore, and this has been enabled now with high throughput sequencing, so we can find those special molecules. So this is a way, again, to modify microRNA and either stimulate their degradation or promote their biogenesis. And this is just to show you that there is a LET7 Lin28 example, and now we know that there is other example as well, but they are not dependent on Lin28. Again, it's another way to do it. Another way to regulate microRNA biogenesis is to affect the genes or the protein associated to their function. This is the Argonaute protein. So Argonaute is associated to the microRNA. This is the one that binds the microRNA at the end and favors the gene regulation. You see, now we know that Argonaute can be modified different ways, hydroxylation, part addition, so poly-ADP ribose, ubiquitination, phosphorylation, and so on. We don't really know much about their function, but we're starting to understand that. So this is another way, again, to regulate microRNA function. Different step now has been discovered, so if you are interested in that, this is a very good review written by American. And in this review, she surveyed or she reported all the different type of modifications that have been found in dicer, DGCRA, Rocha, and so on. All those modifications can somehow affect specific microRNA biogenesis or production or level and so on. So once you have produced the microRNA, it's associated to Argonaute. The next question is what you have to know is how those microRNA can regulate gene expression. So, like I told you about, they're recognizing their sequences or their targets by complementary sequence. In animals, this complementation between the microRNA and its target is not fully the entire length. So there is bulges and mismatches within the complementary sequence. And this is something that we don't see in plants, because in plants, microRNA are fully complementary to their target, but in animals, they're not. So they have bulge and mismatches. So this is what I call, partial complementarity. Most of the region where microRNA are binding is the 3' UTR region of messenger RNA. So this is where you're going to find the microRNA sequence binding site, and all the algorithms that are available right now on the web are mainly focusing on the 3' UTR. We know also that most microRNA need to be base paired with few nucleotides at their 5' end. So this is the messenger RNA, this is the non-coding 3' UTR, and this is 5', 3' polarity. So the microRNA will base pair this way, so this is 5', 3', and there is a region there that we call the seed region, and this region is depending on who's you're reading, it's from nucleotide 2 to 7, 2 to 8. Most of the microRNA need a fully complementarity with their target at this region. The rest doesn't really matter, but we know that this region seems to be important for the specific recognition and the efficient silencing of the target. This is what we call the seed region. Again, I told you this, most of the microRNA, but now there is an example that there is what we call seedless microRNA. Another thing that we have observed is more you have microRNA binding sites at a 3' UTR, more the repression will be effective. So this is cooperativity type of system. So more you bring microRNA and argonauts to a target, more the repression, better the repression will be. And what microRNA are doing, we're going to go into that, but the end of the function is to alter protein synthesis or block the production of proteins from the specific microRNAs, the specific messenger RNAs that is targeted by the microRNA. Because the complementarity is only essential for 6-7 nucleotides long, you can easily see, like you were mentioning, that one single microRNA can target different messenger RNAs. Because the complementarity is always required. And this is something that you have to be careful. So if you are interested to know whether your gene is regulated by a microRNA, one obvious thing that you need to do, and of course, it's not so obvious because it's not telling, no algorithm is really looking at this, but you have to make sure that you go to there is target scan, different ways to look. Okay, I have a gene X, I don't know, RAS, and I want to know if there is any microRNA that regulated this RAS messenger RNA. I just go into the web, I write RAS, and you will have hundreds of microRNA. But one thing you have to make sure is that your target is expressed at the same cell as your microRNA. It looks stupid, but most of those algorithms are not looking at this at all. So you will have a list of 100 microRNAs that can be potentially targeting your gene. But easily what you can do is just looking at their expression in your specific cell types and so on, and you can narrow down to very little amount of microRNA. So this can speed up a lot your search for mutative microRNA that can regulate your gene. Another thing that has been observed and reported is if you extended the complementarity with the target, what's going to happen is you will lead to endonucleotide cleavage. I told you that some organelles have this endonuclease activity through their Pee-wee domain. And if the complementarity expands with the microRNA in its target, so you see here there is a bulge. In most microRNA, this is what we see when they paired with their target. If this is base paired with its target, it will generate direct cleavage of the targets. Why? Because the AGO2, the human AGO2 has the endonuclease activity, and it cleaves at between position 9 and 10 from the microRNA or the short RNA that is associated. So if you have full complementarity with a target, there is a great chance that your target will be cleaved if the microRNA is loaded on human AGO2. Like I said, there is 4 argonautes that bind microRNA in human, AGO1 to AGO4, but only AGO2 can cleave the target. So there is example of this here that has been reported, like in this review article. I know this one is not a review article; it's a research article where they found in different 3' UTRs, they found an extended complementarity with some microRNAs, and this leads to the degradation or the cleavage, the direct cleavage of the target. So what microRNA does in the complex, in fact, through its complementarity with its target, it brings argonautes and the associated protein to the messenger RNA, and this is the argonaute and its associated protein that block or affect protein expression. It's pretty much the same type of system that you have seen probably in your courses before about splicing or the splicing machinery. This is mainly this. It's a ribonucleoprotein complex. It contains the RNA molecule that brings the complex, the protein, at the 5' splice site or 3' splice site. So it's recognition by base pairing. Same thing with microRNA. So why we know that is because if you recruit artificially argonautes to a 3' UTR or a target, this is a reporter, without the presence of microRNA, it's sufficient to repress translation or to block the synthesis of the proteins from that reporter. It shows that just by recruiting argonautes and probably its associated protein to a 3' UTR of a messenger RNA, it's sufficient to block the production of proteins from this messenger RNA. So microRNA is there to just bring this silencing complex to the organism. To the target. Another intriguing thing is how microRNA, with such few complementarity with its target, I told you that the seed region, which is 7 nucleotide long, is sufficient to target the messenger RNA and induce its degradation. How a microRNA in the cytoplasm, for example, can find its targets where you have hundreds and thousands of messenger RNA and other RNAs hanging around putative, target sites. For this by base pair complementarity. So one thing that we have shown is, in fact, the argonaute complex interacts with the ribosome. So ribosome is split for translation, and one subunit of the ribosome called RAC1 is physically interacting with argonautes in human cells and as well as in worms. And what it does, it recruits microRNA at the translation site. This is a way somehow to concentrate microRNA and their silencing complex at location where messenger RNA will be found, which means at the ribosome where the translation occurs. And so this is a way to recruit or concentrate microRNA. Another thing which is quite intriguing, and in fact this slide should go before. So if you look at the literature, you will see different ways microRNA can regulate gene expression. There is many papers that show that the recruitment of microRNA organoid complex with 3' UTR of a target will recruit deadenylate complexes, and this will stimulate degradation of the messenger RNA by deadenylating the messenger RNA and lead it to the degradation. There is also examples where microRNA organoid complex, the miRISC complex, will induce proteolysis, so degradation of the nascent peptide that is produced by the ribosome. And somehow it will stimulate degradation of the peptide that is generated by the ribosome, so you will never see a full-length functional protein. There is also examples where the recruitment of argonauts through microRNA will block the initiation of translation as well as the elongation of translation. So as you can see, there is different ways that have been described in the literature using different systems. Most of them is culture cells, humans, mouse, and so on, where people have shown that the miRISC, the silencing complex that contains argonaute, microRNA, and other proteins, in effect, can lead to complete blocking of protein synthesis from the targeted messenger. So a way to reconcile this, I think it's coming slowly, and there is other data sets that support this. The fact that we have multiple argonautes in organisms, like I told you, in human you have AGO1 to AGO4. I think what we can elude is it depends which argonaute will be associated to your specific microRNA, and the association of argonautes with of one specific argonaute with the microRNA can somehow affect what kind of repression system that will be used by the cell. This group show is the Tomari's group in Japan. They show that if the microRNA is bound by argonaute 1, it will be associated to another protein that we're going to talk about in a few minutes called GW182, and this stimulates degradation of messenger RNA. If the microRNA is bound by argonaute 2, for example, in this case, it will block initiation of transcription. What I'm trying to say here is, in fact, I think all those types of models are probably true, but I think we still don't understand fully what kind of silencing complex is built through microRNA. We don't even know if there is specific silencing complex, argonaute, and other associated proteins that are specifically associated to one microRNA and one cell type and so on. So we need to better understand this to really fully understand how it works. Although some people like to do very strong statement of how microRNA works, and this is one of the issues from the field, because there is very prominent investigators in the field that will do massive analysis of transcription, genome, and so on, and they will come to which conclusion that can be somehow misleading for people not in the field. Like example, this one coming from David Bartels at MIT. He came out with this paper in 2010 where he said that microRNA, the function of microRNA is what? Is to do this, stimulate degradation of messenger RNA. All the other thing is crap. Doesn't really work, it's not really important, and so on. So you come with this data set, people which are not in the field think that this is the way it works. So it's very dangerous because people are, some people, if you look at, is your target is regulated by microRNA, you say, well, it affects the stability of my messenger RNA. So I will make a correlation. Express a microRNA, block a microRNA. If my gene is regulated by this specific microRNA, I should look at the messenger RNA level and see variation. But in fact, it's not true for all. You have to be careful with this. You should always look at the protein level and not only at the messenger RNA. And this is two clear examples of this. So 2 years after, 2 other groups decided to prove that it's not the case. So in this case, what they said, that it's translational repression and it's not degradation of messenger RNA. Using different systems, different things, you know what I mean? So you have to be very careful about this. But one thing that is for sure is all microRNA will lead to a blockage of protein synthesis or production. So if you suspect that your gene is targeted by a microRNA, always look by Western blotting or mass spec or anything, the level of your target. This way it's going to be sure that you have something going on. Because if you only look by RT-PCR, sometimes you're going to see, sometimes you won't, but you can miss something. It's very important to look at. And many people in the field, including me, I'm not very a big fan of this study because they have used overexpressed system, they overexpress many things, microRNA and so on. It's not in the natural setting. And in this case, at least they're using natural setting. The natural level of microRNA, natural level of target to draw their conclusion. I think for me, it's a better way to show the biological significance of something. But I think it's a mix of everything. And this is the other thing also. We also be reflecting, different cellular states. If your cells are dividing, if your cells are differentiating. differentiating. Maybe it's a different way microRNA will regulate gene expression. Some things that you have to keep in mind. Another correlation that has been made with microRNA is their function associated to a specific region in the cytoplasm. So what we call the GW bodies. This has been big when the field had started, like back in the end of 2000. People were saying, okay, this is where microRNA works in the what we call the GW bodies or the P bodies. But in fact, many studies afterward have shown that it's not true. It's not important for microRNA to be specifically associated to this granules to be functional. But one thing for sure that came out from this is this protein called GW182. It's in human. It's called PNRC6. And this is a strong interactor of organelles, and it's required. It's found in these granules, and this is why people have been misled. But we know that the interaction with GW and organelles, in most of the time, it's important for regulation of gene expression. Although there is example now that we're working on that or not. But the thing is, the GW protein, what it does, it recruits the deadenylate complex, CCR4 NOT complex, and stimulate degradation of messenger RNA. So a lot of work of this has been done with many labs in the world, including Alan Sonnenberg that has been working on this for many years, and Mark Fabian that is like LDI. Just started his lab working on this, and Thomas Duchenne as well at Hegel. So this is what GW does. It recognizes and brings this deadenylate complex and stimulate degradation of messenger RNA. So this is one of the ways that microRNA are doing gene regulation. Again, it's very tempting to make global assessment saying that GW are essential for microRNA function. I think again we have to be careful in what stage it's working. Another amazing feature of microRNAs in this is a very nice paper that has been published 5 years ago. And this is very neat because they show that in mouse eyes, if you just put some light, in front of the mouse, what's happening is it will change the regulation by a specific microRNA. So you can turn on and off microRNA regulation just by stimulating, for example, eye with light. So what is very exciting about this is you have to keep in mind that microRNA binds to messenger RNA, block translation, block protein synthesis. If it doesn't degrade my messenger RNA, it will be a way to make a switch on and off on translation. And so you just need to relieve microRNA from the target and start translation again from the same messenger RNA. And this is very rapid because you don't need to go through the transcription, the maturation of free messenger RNA, export, and so on. It's already there, ready to go. And there is more and more example, and this is really associated more to the neurobiology field where we think that, if you know a little bit about this, axon that drives where you have local translation. So most likely microRNA can have a very important role on this. And in this paper, they really clearly neatly show that the same target, the same messenger RNA can be shut down and shut up, and turn on and turn off by a single microRNA without having translation or transcription of the messenger RNA. And there is way also that you can modulate microRNA function. We talked about biogenesis, post-translational modification of key components of the microRNA, but there is a lot of interplays going on that we don't know much about. The 3' non-coding region of a messenger RNA is filled by RNA binding proteins. There is a lot of proteins that bind this region. And we're starting to see interplay and function of those proteins that bind nearby microRNA binding sites and somehow affect the microRNA function. So there is a lot of things going on there, and this is a very exciting place to look at how microRNA can be regulated. This can be condition specific, upon stress, upon light, like I told you, tissue specific, development, and so on. And just to mess up more of the system, there is a way that proteins can expose microRNA binding site by changing the structure of RNA molecules, like the 3' UTRs can have secondary structures, proteins like PUM1 that bound, cell cycles, specific cues in the cell cycles. What's going to happen? It's going to bind the structure, unwind the structure, and now make the site, a binding site available for microRNA. This is another example here, which is like the reverse. Proteins will bind, form a secondary structure, and mask somehow the bindings of the microRNA binding site. And this is also a very neat example where microRNA does not act on protein synthesis per se, but in fact it's expressed to act as a decoy for RNA binding proteins. So there is one example of this, and it's quite neat. So the microRNA is produced, not bound by argonates, but it's bound by another factor. The binding to another factor stimulates the translation of a gene that usually is blocked by the binding of this protein to the messenger RNA. So there is a lot of interplay going on, and I think there is a lot to learn from this. And finally, one thing that you might have learned recently, it's very popular in the field, the circular RNA molecule. So there is, a few years ago we discovered that our cells are loaded with tons of those RNA molecules. They are round, without any end, and they are populating our cells. We don't really know what they do, and and so on. And there is a lot of people that think that probably circular RNA can be a good way to quench or control the microRNA bindings to their target, because they can be natural, binders for microRNA sequences, and somehow act on their function. And this is very exciting because a few years ago there is a group in the US that has been pushing a lot the fact that maybe non-coding RNAs can act as a decoy for microRNA, so the expressing are non-coding RNAs. But many people afterward have shown that it's almost impossible because the level of long non-coding RNAs that you can have in the cell is very low compared to the level of some microRNAs. So it's really hard to believe that they can quench. But this is the nice thing with circular RNAs, that they are expressed like massively in the cell, so it might be a natural way to regulate or to change the balance of microRNA in some target. So if I sum up this, basically it's microRNA are mainly produced by RNA fold 2, so they are subject to gene regulation like any other protein coding gene. The first maturation occurs in the nucleus, the microprocessor, that we call a microprocessor. Second maturation in the cytoplasm with the by the dicer complex. They form an active complex. You have the microRNA, an argonate, an accessory protein like GW182 and other proteins that we and others are looking at actively on this. In animals, they bind mostly the non-coding region, the 3' non-coding region of messenger RNA, and need to complete shutdown of protein production in different ways. So, there is still many things that we need to address in this field. The target of microRNA is a big thing. It's really hard to do because the complementarity is not full. So it's very hard to find targets based on 6, 7, 8 nucleotide-based pairing. So there is a lot of work to do about this. I think that is the best way to do it. Is, people in different fields are starting to look at microRNA regulation and their favorite genes. This is the way we're gonna be able to go over that challenge. How the microRNA pathway control. This is something also that is very that people are working hard on. How microRNA regulates expression, trying to really find a way to understand clearly how it works, cell-specific, cell-type specific, and development, and so on. And this is also an exciting field right now where, how microRNA can travel from cell to cell in active trans. And this is something that, like I told you, the paper that just came out a few days ago in Nature that I think really showed that there is something going on there. So, has been, circulating now in the field, but, there is no clear proof of this besides, like I said, the recent paper that seems very that's about it. So this is other reviews if you're interested to read. It's the ones that I put also on my slide. This is good things to look at. And do not hesitate if you have any questions or comments on the class. If it's too boring, too long, too stressful, too anything, just send me an email with your unknown or anonymous Gmail account if you like.